Today I'm going to be presenting my project that I worked for 6 weeks, which is isolation and purification of Azadirachtin from neem oil and evaluation of its antimicrobial antibiofilm effects against gram-positive and gram-negative pathogens. So first of all, the motivation for this project came from the growing problem of antimicrobial resistance, which is increasing interest in plant derivative compounds as like a possible alternative to conventional antibiotics. Neem, also known as Azadirachta indica, has long been used as traditional medicines and oral hygiene, and it contains many bioactive compounds. One of the best known is Azadirachtin, which is widely studied and for insecticidal activity, but its purified antibacterial activity is very less understood than that of the crude neem extract. So the goal of my study was to isolate and purify azadirachtin from neem oil and test its effects against three bacteria. First one is S. mutans, which is a very important dental bacteria, which forms oral biofilms. Second, Streptococcus aureus, Staphylococcus, which is highly versatile pathogen causing diseases ranging from skin infections or life-threatening pneumonia or endocarditis. And third, E. coli, which is gram-negative bacteria, also known for food poisoning and UTI infections. So my hypothesis for my project was that purified azadirachtin would reduce both bacterial viability and biofilm formation in these species. For my methods, azadirachtin was extracted from neem oil using methanol water partitioning, followed by hexane defatting. The extract then was concentrated, reconstituted, and analyzed by UPLC to confirm the presence of azadirachtin by comparing it to the standard. And antimicrobial testing was then carried out using four approaches basically: MIC, minimum inhibitory concentration; MBC, minimum bactericidal concentration; and agar-well diffusion; and then at last biofilm inhibition assay using crystal violet staining. All three bacterial species were tested, and the data was analyzed mainly using Creutzfeldt-Wallis test because of sample size being small. Starting with UPLC results, azadirachtin was successfully detected in all three extracts with retention time about 5.3 minutes, matching the standard. The mean azadirachtin concentration was about 5.9 plus minus 0.19 microgram per ml. These reconstituted extracts were then used as neat stock solution in the biological assays. My in the agar-based inhibition assay, neat azadirachtin showed a clear antibacterial activity against all three species, which the inhibition zones having 14.0 plus minus 1 millimeter for S. aureus, 13 millimeter for S. mutans, 13 millimeter for E. coli. These values were close to antibiotic control as well, while the vehicle control produced no inhibition. The 2x dilution showed a very weak and a little inconsistent activity, but it showed some around 3 millimeter range of inhibitions in some replicates. Overall, this suggested that azadirachtin has measurable antibacterial effects at the neat concentration against both gram-positive and gram-negative bacteria. In the MIC assay, azadirachtin showed a concentration-dependent inhibitory effect. The strongest inhibition being like two highest concentration, 5.9 and 2.952 microgram per ml. For example, E. coli showed for E. coli, growth was reduced to 17.98% at the highest concentration, and S. aureus it was 21.02%. As the concentration decreased, growth increased, and the lower the concentration often rose above the untreated control. Statistically, azadirachtin concentration had a significant effect on E. coli and S. aureus, but not S. mutans by Creutzfeldt-Wallis analysis. MIC results were more variable. For E. coli, no growth was seen on one replicate and at the highest concentration, suggesting possible bactericidal activity. For S. aureus, growth was seen in all tested concentrations. For S. mutants, results were a little inconsistent between the replicates, showing that the inocula taken from the well grew on my agar plates. Sorry. For biofilm inhibition, all three species responded, but in different ways. As Oreo showed a little stronger inhibition at about like 2.95 microgram per ml, where biofilm was reduced by 55.52%. For S mutants, strongest inhibition was at 1.47 microgram per ml with 57.12 inhibition. But interestingly, the highest concentration actually increased biofilm formation relative to untreated control. um, E. coli showed the most consistent pattern with inhibition strongest at intermediate concentration and declining as the concentration decreased. So overall, azadirachtin had the species-dependent antimicrobial effect. In the bigger picture, these results suggest that purified azadirachtin contribute to the antimicrobial activity associated with Neem, and the effect is not due to crude extract. It shows that both gram-negative and positive bacteria, which suggest degree of broad spectrum potential. At the same time, response-dependent manners based strongly on both species and concentration, especially for biofilm. So to conclude, purified azadirachtin isolated from neem oil showed measurable bacterial and antibiofilm activity against S mutants, S. aureus, and E. coli, the strongest effect being seen in highest or intermediate concentrations. And overall, the compound appeared to act mainly on a bacteriostatic agent under these conditions. These findings supports my hypothesis and as and promising and azadirachtin being promising plant derivative antimicrobial lead for further study. Thank you very much for listening. Have a nice day.